All right, good morning, class. Um, we are starting chapter 29. Around 10, I got some calls from one or two classmates confirming our schedule for this week. And like I said, today is chapter 29, and the week 4 lab quiz has been open since yesterday. And I will bring out some practice tests for you for the week 4 exam. One question was the grade that, for example, she had, a classmate of yours, she's having an 85%, and she said right now she's missing one exam, and she feels that she's doing well already at 85. So I told her it's 85 because it's just a weighted total, but in the end, it will sum up everything, a running total, and that 85 is not really your last grade or your final grade. The missing exam will pull that to, I don't know how much, on an approximate, probably pull it down by 10 even, depending on how much you would have gotten on the week 4 exam, that being the last final exam. So the bottom line is what the grade you're seeing now is a grade showing you your accomplishments, but you got to finish the exams, or the requirements, because when it sees the requirements as not done, it will consider it as zero. So you have to finish it in order to get the right total that you deserve, right? And also, if you don't finish the major exams, then we don't have a fair estimate of how they finish the class. So um, make sure to finish all requirements and take note that this our class will stop on Saturday, huh? And if you have anything else scheduled, like doing your week 4 exam or finishing up discussions, make sure that you uh, stick to your to-do list. We have to pass ANP1 before we can enter ANP2. Huh? It's a prerequisite. All right. So if you have any questions, um, email me, text me, and I will get with you and help you sort out your question. Okay. All right. Let's go ahead and start with chapter 29. Chapter 29 is about the embryo, the pre-embryonic and the embryonic stage. It also talks about the other systems. Chapter 29 will start on some other system, which I think will be repeated in the other when we get to ANP2 anyway. So I will go ahead and focus on the embryo and let you understand, especially the capacitation, huh? And this will lead us to the developing embryo in the chapters following this. Okay, so embryology is the study of prenatal development. Huh? By the way, NATO, NAT/O, means birth. Huh? So if it's natal, means pertaining to birth. If it's prenatal, pertaining to before birth. Uh, Perinatal is pertaining to within birth. Peri means within. And postnatal means pertaining to after birth. Now, on this point, we would like to know the process of sperm migration, how an egg prevents fertilization by more than one sperm. It has only to be one sperm, and then also how it formed to a fertilized egg. Huh? Now, in this topic, the embryo may be considered to have different meanings, but it is like the stage before the fetus. It is the stage after the blastocysts. So many embryonic terminologies. Now, in an essence, The embryo is one that is 16 days old and has the primary germ layers. The primary germ layers are the ectoderm, mesoderm, and endoderm. Huh? And correct that, please. That should be endoderm. And this leads up to the event of embryogenesis. Now, when ovulation took place, which is at the 14th day, uh, the egg has been released and it is viable for a maximum of 24 hours. So, if we are wanting to have a union or fertilization to occur, the sperm should be within that 24 hours. Okay, the sperm can be have been there. The sperm has about 36 hours viable time, huh? 36 hours. So it could have been there a day before, right? And then was still there when ovulation started. It could have been there within the 24 hours, or it could be there at the last part of the 24 hours. So this is the window that the male and the sperm has in order to result in a fertilization. Now, I would also mention if a couple, a man and a woman, is desiring to bear child, then they should pay attention to this 24 hours of the egg and 36 hours of the sperm, huh? And put it on and map it out. You know, they measure the temperature of the body, they do the spin Barcade test, the mucus of the cervix, all pointing towards that day of ovulation. And when they do it properly, they may be able to be successful. On the other hand, if uh, having a child is not yet in their plans and they want to avoid it, then they should pay attention to not get together when that ovulation happens. So you want to make sure that it's clear and there's no chance that the sperm is in the area. Okay, so those are very important things to remember, no, depending on what their plan is. Okay, now once the female has ovulated and the sperm is now in the cervical area, it will try to go up the uterus, but there's many reasons why the sperm may not make it to the egg. One, the vaginal acidity environment is really strong and can destroy the sperm. The the fact that it has to go up sometimes is a factor. There are therapies that require the female to be in a certain position to aid the sperm by gravity to get to the fundic area and into the fallopian tube, and may be there for about 30 minutes sometimes. Second is there's a cervical mucus plug, right, that they have to penetrate through. So that's another hindrance. There's white blood cells in the uterus that if they encounter, they will get destroyed. The sperms will get destroyed. And if the ovulation took place in one ovary, but most of the sperm went to the other side, then again, it's not gonna happen, right? Sometimes this is one of the reasons why no pregnancy ensues. Of the 300 million that were put in the cervical area, about 200 spermatozoa reach the vicinity of the egg. Imagine that 300 million and about 200 makes it to the vicinity of the egg. There's a high mortality, huh? With all the hindrances that are put upon uh, the sperms on their way. The sperm move by lashing their tail, their flagella, as it moves through the mucosa. The cervical mucus guides them through the cervical canal, and nutrient contractions pull up the seminal quantity and spreads it through the uterus. It helps physiologically. And then the chemical attractant molecules released by the egg attracts the sperm from a good distance when you're close to it. Again, the sperm, right, can reach the area where the egg is in about maybe 10 or so minutes. But penetrating the egg actually takes about 10 hours. So you're in the vicinity and the egg is there, but it takes about 10 hours for the sperm to successfully penetrate the egg. This is called capacitation, the process that migrating sperm must undergo to penetrate the egg. And there's many reasons for the successful penetration. Sperm is viable for, like, some say 36 hours, some say even 6 days. 36 hours, I think, is like an average. So we have to be sure that both items are in place in order to have a successful fertilization. When that happens, the sperm will try to penetrate the shell or the cover of the egg, and it may not be penetrated right away. There may be the first line of sperms that will try it, fail, and then the second line picks up to where they have finished. And so finally, the successful one will make it through inside. The sperm applies hyaluronidase and acrosine enzymes in order to do this. When a path has been cleared, a sperm binds to the zona pellucida and releases its enzyme to digest some more pathway. The sperm head and meat piece enters the egg. The egg destroys the sperm mitochondria, which it doesn't need now, and then passes only maternal mitochondria onto the offspring. Right? What it needs from the sperm is the 23 chromosomes. The 23 chromosomes of the sperm will be joined by the 23 chromosomes of the egg, making 46 chromosomes or 23 sets. Polyspermy is fertilization by two or more sperms, which will produce a doomed fertilized egg. It has to be 23 pairs. Remember, even if you produce one more chromosome, it can result in a defect already. What more if you have an extra 23 chromosomes messing up the pairing? So this will end up in a failed egg. There's two mechanisms to prevent polyspermy. There's a fast block and there's a slow block. Fast block uses sodium channels in the egg membrane. The inflow of the sodium depolarizes the wall and creates a barrier for the next sperm to go in. So once the first sperm gets in, this fast block initiates and prevents the other sperm from getting in. All right. There's another method. It's called the slow block method. And what happens is when the sperm penetrates the egg, the wall itself releases an inflow of calcium. This will end up in granulation, thickening of the wall, so no other sperm can penetrate after the successful sperm. Right. Again, this is to prevent polyspermy. Right. We only need one set of 23 chromosome pairs. So adding another sperm will complicate the union. This picture shows you how one sperm has done some good digging, if you would, and if he's not successful, another sperm behind him will try to follow through, and the successful one will finally break the wall, if you would, and may be able to get in. After which, this one shows how this one was successful. After which, thickening of the membrane, granulation of the membrane will happen, and no other sperm will make it. See, this one is being deflected already. That means this has already been accomplished. One sperm and one egg. Okay, Look at all those sperms trying to get in. So there's only one lucky sperm. Meiosis 2 is when the secondary oocyte begins with this process. The sperm and the nuclei swell and become a pronuclei. The fertilized egg will now be called a zygote and will go through meiosis. The course of pregnancy is divided into 3 trimesters, the first, second, and third. The first is from the fertilization up to 12 weeks. So this can happen within 24 hours, and from there it starts on its first trimester up to 12 weeks. This is more like a make or break trimester, because if the sperm and the egg provided a good union, this would result in a successful uh, embryo. If not, this is also the time when it will not really make it, which in some cases may be better because you don't want an embryo that's not completely finished. Remember that after fertilization, they will still have to travel all the way to the uterus to implant. That's part of the 12 weeks. Conceptus, which is the product of the fertilization, or the product of the union of the sperm and the cell and the egg, is vulnerable to medications, nutritional deficiencies, and other factors like maybe some stress or even viruses that can prevent development at this point. So there's a lot of factors against it. To be able to go through this means that this egg, mature egg, fertilized egg, is really strong enough. The second trimester starts from week 13 to 24. The organs are complete, and most of them are just finishing up their development, complete in the sense that they are anatomically formed already, and they're just finishing up physiologically to be active. The fetus looks distinctly human, and we can see this also in ultrasounds. And it's approaching the third trimester. Now, the third trimester is 25 weeks up to the time of birth. And at this point, the main goal is to mature the organs, grow accordingly, and be able to sustain its life at time of birth. If you have gained weight of about 5.5 pounds at 35 weeks, then the fetus, now called the fetus, is considered to be viable, huh? And also better if we at least cross the 35 weeks. There are cases when they are born prematurely, and of course there are some dangers to that condition as well. The pre-embryonic stage is the first 16 days of development up to the stage of the embryo. It involves cleavage, implantation, embryogenesis. We'll cover that in this chapter. Let me mention first the situations when you have twins. We have two types of twins, a dizygotic and a monozygotic twin. Dizygotic considered as the paternal twin, and monozygotic the maternal twin. So how does that happen? Dizygotic is when there's actually two eggs that were produced during ovulation. Huh? Two eggs. It may be one ovary produce one egg, and the other ovary produce another egg. So this is more common, huh? Two-thirds of twins. So what happens is, because you have two eggs, then you have viable two individuals that can be brought up. So two successful sperms will penetrate the two eggs separately, huh? Still one egg, one sperm. But then we would have two fertilized eggs. They will go to the uterus, both of them, and they will implant separately, huh? They will implant separately, and therefore will have their own placenta. That's a paternal or dizygotic twin. The maternal twin comes out of one egg. So there's actually one egg that was fertilized, huh? One egg, one sperm. But what happened is the embryoblast divided into two. And with that, it ends up you're able to form two entities or two human embryo or two human embryoblast. Now, genetically, this is very identical, huh? They may be of the same sex and very similar. This is the best chance for transplant because they almost have the same tissue, huh? So monozygotic twins are the best for transplants or sometimes for donor cases. For example, the other one needed a kidney, um, and one of the twin wants to donate his kidney. That's one of the most perfect matches, if not the best. Okay, huh? Monozygotic twins. Here's a dizygotic twin example where they have their own placenta and they came from two eggs. Okay, cleavage, as we mentioned a while ago, is a division between the pre-embryonic stage, happening in the third day. Huh? Now, cleavage would end up about 30 hours after fertilization, and it will go through many stages. The marula stage would be the stage when you first enter the uterus, and then it becomes a blastocyst at the same time. The blastocyst stage is the one that will implant in the uterus. So in this picture, you can see the egg cell, all right, coming out, being from ovulation, and the sperm cells will try to unite with the egg cell. When one successful sperm has penetrated, it will then start its way through the process. It will become a zygote, a 2, a 4, and 8 cell stage zygote. Then it becomes a morula as it enters the uterus, as you see here. Huh? So it's traveling at the same time it's traveling in the fallopian tube, it's continually developing. Huh? Upon entering the uterus after 72 hours, it's called a morula. Then it changes into a blastocyst, right? The blastocyst is the form that implants in the endometrium, and that's about 6 days. Huh? So within 6 days, something is happening already. It's less than a week. And hopefully, the blastocyst would implant good enough and deep enough in the endometrium. Implantation, the blastocyst attaches to the uterine wall and then continues to bury itself, to dig itself in to a good layer. By doing that, it's able to sustain itself, and you'll have enough layers to provide good nourishment to the um, embryo. Syncytial trophoblast will grow into the uterus. It will be the beginning connection with the endometrium. The endometrium reacts to this growing entity by covering it all around. So in about a week's time, implantation would have been started. And at this point, the menstruation will not ensue because it will trigger chemical reactions in the uterus telling it that it will go into pregnancy instead. Trophoblast secretes HCG, and HCG will be behind the female or the mother's system. In fact, this is one that we can measure as a sign of pregnancy. Progesterone will be high at this point, which at the same time suppresses menstruation. No cramping of the uterus. HCG levels rise in the mother's blood until the second month, and it will take it all the way to the beginning of the third month. At this point, the corpus luteum is making HCG. The corpus luteum is providing progesterone and other hormones, waiting for placenta to take effect. You can see the how the blastocyst finds its way to sink in, the syncytial trophoblast starting to connect the blastocyst to the endometrium, and then finally making a good implantation. See, this is the surface, and the entity has sunk beneath the surface, which means this blastocyst is on its way to being safe. Huh? Embryogenesis is arrangement of blastomeres into three germ layers: ecto, meso, and endoderm. And for this, the layers will end up as such. You have the endoderm on top, the mesoderm. This is the endoderm, a little bit on the lower part. And then you have the mesoderm, and then you have the ectoderm. All right. A primitive streak will start developing in the area, running down its middle like a groove, and then it will be bilaterally symmetrical. This will be the future right side, dorsal and ventral surfaces, and cephalic and caudal ends, because this will curve in, as you will see later on. Multiple epiblast cells migrate into primitive groove, and in a few days, epiblast will be forming a third layer, the mesoderm. The remaining epiblast remains as the ectoderm. Of these layers, the mesoderm gives rise to the muscle, bone, blood from the mesoderm. And what happens is the next line, the ectoderm, gives rise to the integumentary system and even the nervous system. If there was any situation where the fertilized egg did not make it to the uterus, anywhere that it did not make it to the uterus, you consider it as an ectopic pregnancy. The blastocyst form will implant somewhere other than the uterus. Sometimes it actually goes into the abdominal area, which is kind of rare. Now, some of the sometimes it gets trapped in the fallopian tube. We call it tubal pregnancy, right? It implants in the uterine tube. Now, this can also be called as an ectopic because it's outside of the uterus. It's just one example of ectopic pregnancy. The problem is, as it continues to grow bigger, it will rupture the tube plus minus 12 weeks. So, and of course, this will be intense pain, which leads us to discovering that it was a tubal pregnancy. It's hard to determine it until it makes the pain is felt, because we will have assumed it went to the uterus. Abdominal pregnancy implantation happening in abdominal pelvic cavity is 1 out of 7,000, which actually is life-threatening. So that's another complication of this. In embryonic stage, the flat disc is converted into cylindrical form. This happens in week 4. A yolk sac is formed and the embryo becomes like a C-shaped. So further on, what will happen is folding will occur. The entire surface is covered with ectoderm, and it will form both cephalic and caudal, and on left and right sides. In fact, it's what we call embryonic folding. This is the length of the embryo. It will fold inwards on the caudal and cephalic sides, right? And that's what you see on the longitudinal picture, right? It will go towards this final form. Meanwhile, the sides should be seen here. This is cross section. The sides will also fold inwards. Ultimately, it should look like this stage in 30 days. You will see the beginning lung, we call it lung bud, liver bud, right? The heart area is already uh, starting there to form. This is the primitive gut. This line maybe it will end up be part of the nervous system. So it's on its way in 30 days. That's the first month. From here, you can see the development of the germ layers. The ectoderm gives rise to the epidermis, the skin, the nervous system, and much of the visual, and part of the inner ear. The mesoderm, like we said, gave rise to blood, muscle, also skeleton, all the way to lymphatic tissue, gonads, kidney, and ureters, cartilage even. And the endoderm gives rise to the gut, respiratory epithelium, urinary um, organs as well. Here's a picture of the developing human, huh? From the first enfolding that we saw into the future embryo and the future fetus, huh? Very interesting. Accessory organs develop with the embryo, which is the placenta, umbilical cord, embryonic membranes, amnion, all the way to chorion. Huh? Amnion is a transparent sac that covers the entity. It grows to completely enclose the embryo or the entity. Huh? It will fill with amniotic fluid. This mechanism where you have amniotic fluid in the amnion will protect the embryo from sudden trauma. It can also be anti-infection or prevent infection from coming in. The temperature is maintained inside. Growth is allowed to develop inside with no restriction. Huh? Very important for the development of the fetus and later on. Huh? Um, this will also prevent parts of the embryo from being in close contact with the mother's body. We don't want any adherence. So this protects the embryo in that safe environment. Okay. There's a situation when the obstetric doctor will advise the couple if they want to have amniotic fluid analysis or amniocentesis. This is done if there's a strong chance of anomaly that may happen. So by checking the amniotic fluid, they hopefully will identify if an anomaly is present or might happen. Yolk sac will form, rising from the hypoblast, and then it will contribute to the formation of the gastrointestinal tract, blood cells, and future egg or sperm cells from the yolk sac. The allantois also will form an outpocketing of the yolk sac. This is the foundation for the umbilical cord and will also be involved in the formation of the urinary bladder. Chorion is the outermost membrane that has connections to the chorionic villi and the entire surface around it. Now, the villus chorion forms fetal portion of the placenta, of the future placenta. So as you see, what was a small implantation is now a major entity in the endometrium. See, huh? This is a picture of the future placenta connected to the umbilical cord. Okay, huh? And this comes after the child is born. During pregnancy, the conceptus is nourished in three different ways through the uterine milk, if you call it, through trophoblastic nutrition, the one that's around the actual entity, and placental nutrition. Uterine milk is high in glycogen, and the conceptus absorbs this fluid as it goes down the tuba and lies free in the uterine cavity. Well, the trophoblastic nutrition helps to provide additional nourishment. Progesterone from the corpus luteum stimulates these cells to proliferate and therefore adds a store of glycogen, protein, and lipids. The deeper the conceptus burrows into the endometrium, the more in situ trophoblast is able to connect with it. Then this is actually going to supply the embryo for the first week of implantation. Now, the placental nutrition diffuses from the mother's blood through the placenta into the fetal blood, carrying with it oxygenated blood, nutrition, even antibodies. So this will be a good overall nutrition for the fetus or the embryo. So if you see it, trophoblastic nutrition starts a little bit on day 0 and then gets stronger as it shows here, but it weans and tapers at the end about within 12 weeks. Meanwhile, placental nutrition would have started at the fourth week and get stronger and stronger, and actually widely uh, provided all throughout the pregnancy, placental nutrition. So you can see there's good coverage from beginning to end until the time of birth. Placentation is a development of the placenta in terminology. Chorionic villi are extensions of syncytial trophoblast that help to stabilize the fetus. Now, mesoderm grows into the villi and gives rise to blood vessels connected to the embryo. Blood vessels are important because through the umbilical cord, you will have the incoming good blood or oxygenated blood and the outgoing deoxygenated blood or where maybe waste material from the fetus. The umbilical cord contains vessels, There's two umbilical arteries and one umbilical vein, Two umbilical arteries, one umbilical vein. The umbilical cord is connected to the fetal heart. Blood flows into the placenta by way of the umbilical arteries and then returns to the fetus by way of the umbilical vein, So going out, okay, is by the umbilical arteries. Going to the fetus is the umbilical vein. The umbilical arteries, just like the pulmonary artery, is known to carry deoxygenated blood. This is two vessels or two types of vessels in the human body that is known to have to have deoxygenated blood. We will talk more about the pulmonary artery when we get to the circulatory system, but they are known for, and in fact, they're favorite questions in the board exams. If ever you see them as choices, make sure to remember that the pulmonary artery and the umbilical artery have deoxygenated blood. Having said that, pulmonary veins and umbilical veins have oxygenated blood, right? I have oxygenated blood. This is a bag of odds, a major bag of odds in our listing. As the placenta grows, the villi grows and branches inside, and oxygenated nutrition is passed to the fetus. Meanwhile, fetal waste is passed on to the mother for disposal, right? And once in the mother's blood, this will be part of what the kidney will filter out. It is also uh, known that the placenta can be permeated by nicotine, alcohol, and other drugs, which means it can affect the fetus. By the end of 8 weeks, that's about 2 months, all organ systems are present. The conceptus is about 3 centimeters long now, and at this time frame, considered as a fetus, from an embryo to a fetus. Bones may begin to calcify, and you will have the heart, okay, beating since week 4, now being more consistent. It will now start to circulate blood. The heart and liver are large, which is probably one of the bigger organs at this stage. The head is nearly half the total body length, which is expected at this stage. The fetus is in final stage of prenatal development. At this point, we are trying to develop and grow it stronger. The umbilical placental circuit is active and is the main source of nutrient for the fetus. The internal iliac artery gives rise to the two umbilical arteries. This blood is low in oxygen and high in carbon dioxide. The umbilical artery blood discharges waste in the placenta, like we said, while the umbilical vein carries oxygen and nutrients, which is brought to the fetus. The umbilical vein carries venous blood through the liver to nourish it. immature liver not capable of performing it, but it will end up maturing through the months to follow. And the venous blood may bypass the liver, what we call the ductus venosus. And then eventually in the inferior vena cava, the placental blood mixes with the fetus venous blood and flows to the right atrium of the heart. Okay, this is a picture of the fetal flow. This is in your textbook, which shows you. Remember, the lung is not yet used at this time. There's no breathing at this point. So in order to make it efficient, blood is redirected back towards the heart and continues on with the other parts of the fetus. Much of this will change when the fetus gets his first breath of air. Like there's foramina vale that is open on both the right and left atrium during the fetal period. Once the fetus is able to breathe, blood is directed to the lungs already, and that hole ceases to exist. It closes. If it doesn't close, it becomes what we know as when a patient says, they said, I have a hole in my heart. That's basically what it is. The ductus arteriosus also becomes closed and does not and redirects blood to the lungs going there. Huh? All right. At this point, we'll take a break. Okay, and then we'll come back in a few minutes.